Good morning, everyone. Thank you very much for this opportunity. I'm very excited to talk about my research here in such a prestigious conference. And as it was said before, I'd like to talk to you a little bit about milk and microbiome. So, as an introduction, we know that mastitis is a very important disease, and we have to do our best to try to treat it and prevent it in the best way possible. And I'd like to introduce you to the concept of microbiome. Microbiome is the study of the microbial population, not only involving the bacteria that we can culture on a plate, which is only 1% of all the bacteria existing so far. But we look at the DNA sequences, and through that, we are able to identify all types of bacteria that are present in a milk sample, and what is the relative abundance, which type of bacteria is dominating a certain environment. And with that, we can calculate measures of bacterial load and diversity. So the objectives of our study were to describe the microbiome in the milk of dairy cows with mastitis associated with gram-negative or culture-negative aerobic results. We'd like to determine the bacterial load based on qPCR and compare the microbial populations, again, the whole community of microbial microbes that is present in a milk sample, between affected and healthy quarters. And our third objective would be if assess the effect of intramammary treatment. In this case, we used extended therapy with ceftiofur hydrochloride for 5 days on the milk microbiome, bacterial load, and clinical cure. So our enrollment criteria, in order to be able to be enrolled in the study, the cows had to have clinical mastitis caused by a gram-negative or culture-negative, and they could not have had any intramammary treatment on the 14 days preceding these intramammary infections. Clinical cure and bacterial cure is absence of clinical signs or the pathogen identified on day 0 on both samplings and the clinical scores on days 10 and 14. So this is an overview of our experimental design. It lasted 14 days of sample collection. And on day 0, the cows were identified as mastitic through farm personnel. A sample was collected, and on-farm chromogenic culture was used to identify these cows. I'm going to be talking about this on the poster session later if you're interested. And the same milk sample was sent to a reference laboratory for confirmation of results. We had 80 cows in total, and the way the randomly allocation was performed was only mastitic quarters were assigned to receive either treatment or control. And in every sampling point, we also collected a ipsilateral healthy quarter from the same cow for comparison purposes. As I said before, the experiment lasted a total of 14 days. On day 1, we performed the random allocation clinical score. This is the breakdown between our negative bacteria. We found gram-negative bacteria. We found E. coli, Klebsiellas, and Pseudomonas. E. coli represented the big majority of it, and therefore I'm going to only focus on their results. And every sampling point, we collected samples from the mastitic and the healthy quarters immediately before administration of intramammary treatment. On day 1, the DNA extracted from the milk was used to perform 16S RNA sequencing and also assess bacterial load through qPCR. The treatment lasted a total of 5 days, again with the sample being collected immediately before infusion of intramammary treatment, and in every time point, we collected a mastitic and healthy sample. A 16S RNA was performed throughout all time points, and qPCR was performed on days 1, 3, 8, and 14. On days 10 and 14, we performed the clinical score and collected an aliquot of the mastitic quarter and sent again to the reference laboratory for bacteriological cure analysis. The last day of the study was day 14, and the cows had the last sample collected, and we also assessed the bacterial load on the last sampling day. This is again a little overview of the study. The methods. Microbiome was assessed using Illumina technology. So briefly, we extract DNA, we target the 16S gene, which is like the fingerprint of the bacteria that tells who is in a sample, through 16S targeted barcoded 16S PCR. Prepare the library using kits V2 for 300 cycles on the Illumina sequencer, and this comprised a total of 6 runs and 1,400 samples. Data analysis was performed through the software Quantitative Insights into Microbial Ecology, which gives us classifications from the field all the way to species level, and I'm gonna talk about my results on the family levels. Statistical analyses were performed using JUMP and SAS softwares. Quantification of 16S RNA was used as a proxy of a measurement of total bacterial load, and we did that using qPCR, and as a standard, we used a plasmid that contained the same, another region of the same gene. We looked at the data in a couple different ways, depending on the type of data we had. So clinical and bacteriological cure were analyzed using contingency tables, and we compared ceftiofur versus control. qPCR data and the metagenomic data, such as pathogen relative abundance and bacterial diversity, were analyzed through repeated measures ANOVA with teeth nested in cow as a random effect. And we also did another analysis with the metagenomic data, which is called response screening analysis. This was adjusted for false discovery rate, and basically what it does is it asks, is this family of bacteria associated with mastitis or healthy quarter? And the test, we're gonna run the test, it's gonna say yes or no, and we correct the p-values using the false discovery rate. So I'm moving on to my results. First, I would like to talk about the clinicals and bacteriological cure. I'd like to highlight that we did not have enough numbers to make any statistical inference here, but if you can pay attention to the means of cures between control and ceftiofur, the means of cures on day 10 and 14 for any type of pathogen identified were very similar, and our p-values are big. E. coli results. So this first graph is pretty much descriptive statistics of metagenome. We took the 25 most abundant bacteria, which are ordered from most abundant to least abundant on the left-hand side column. And here we're depicting throughout all the sampling days of the study who dominated the microbiome, with green representing the lowest relative abundance and red representing the greatest relative abundance. And as we can see, the mastitis quarters, both control and ceftiofur, were dominated by Enterobacteriaceae, and I'd like to call your attention for the fact that both scales are different because the Enterobacteriaceae dominated so much mastitis quarters that if we use the same scale, you could not see any shades on the healthy quarters, which indicates that they are much more evenly distributed. And this little picture on the corner depicts the results of the response screening analysis. And then what I'd like you guys to see from here is that the y-axis represents the log worth of the p-value after correction, and the red ball is Enterobacteriaceae, so it's extremely highly associated with mastitis quarters, because the x-axis, the more to the right side of the zero, it's increased on mastitis quarters, and the more to the left side of the zero is increased in healthy quarters. The dotted line in that graph represents a p-value of 0.01. Bacterial load and diversity, we were only able to identify treatment effect on day 3. Treatment significantly decreased the total number of 16S RNA copies, but we could not observe any effect by days 8 or 14. Bacterial diversity here is calculated as a Shannon index, which basically takes into account all the types of bacteria that are present in a sample and how evenly distributed they are within that sample. And as we can see, healthy samples were always much more diverse than mastitic samples, and we cannot identify any statistical difference between treatment and control. By the way, every time you guys see a red or a blue line, red represents mastitic, blue represents healthy quarters. So we asked the question, so does the microbiome of these cows that were affected with E. coli, do they have anything different between the cows that get cured or not get cured? Because we already saw that treatment does not make a big effect. So first, on the red graph, we looked at the rate of pathogen decrease. This is Enterobacteriaceae relative abundance. The dotted line is it's the treatment, and the solid line is control. And we cannot see any difference. Moving on, we would like to see, do cows that get cured or not differ in the matter of pathogen load at the beginning of the study or throughout time? And does the diversity changes? And as we can see, they do not start with different diversities of bacterial loads. Cows that got cured were represented as gray bars, and cows that did not get cured are represented as black bars. And what we observed was a diverging diversity levels throughout time, and cows that could not experience clinical cure had a sustained increased Enterobacteriaceae levels. Now I'd like to move on to our culture negative results. In the same fashion as I represented before, in culture negative mastitis, we cannot identify any striking bacterial group that's dominated the mastitic quarters. And I'd like to call your attention to the little graph on the right. The dotted line represents a p-value of 0.05, and the take-home message here is bacteria are all over the place. Nobody's statistically significantly associated with either healthy or mastitic quarters. However, mastitic quarters that were culture negative had significantly increased bacterial load, although they did not have any bacterial family dominating them. Bacterial load was increased throughout the first samplings of the sampling days, and upon the last day of the study, they approached the healthy quarters bacterial load. Again, microbial diversity was increased in healthy quarters when compared to mastitic quarters, which can indicate that although no bacterial family was dominating, some sort of dysbiosis might be occurring. So with that, I'd like to conclude that mastitic differed from healthy milk in both microbial profile and diversity levels for both E. coli and culture negative. However, culture negative mastitis samples were not found to be dominated by any type of bacterial family. E. coli samples were extremely dominated by Enterobacteriaceae. We were not able to see any striking treatment effect in either bacterial load, rate of pathogen clearance, or cure. Diversity correlated with disease, with healthy samples being more diverse than mastitic samples regardless of the pathogen that was causing mastitis. And we could not discriminate treated versus untreated quarters, or upon the end of the study, we could not discriminate mastitic versus healthy quarters when we performed multivariate analysis. I'd like to acknowledge all my mentors, Dr. Rodrigo Bicalho, Dr. Rafael Bisinotto, Dr. Georgios Oikonomou, Dr. Inta Shuken, all the farm workers, QMPS Laboratory, Cornell University, the Brazilian government for the Science Without Borders project, USDA, and NIFA. And with that, I'd like to thank you, and I'll be happy to entertain any questions.
Thank you, Dr. Ganda. We perhaps have time for one very quick question from the floor. Otherwise, being mindful of time, and there's one question here quickly at the back.
Hello, Simon Dufour from Canada. Thank you for the excellent presentation. I had a question regarding the negative culture results. That shift in diversity that you see, despite the fact that no pathogen were isolated. What are your thoughts about this? Is it that there was a pathogen that is not there anymore, and it induced a shift in diversity in the microbiome that we still see after the clinical mastitis? Or is it that we have perhaps a pathogen that is still there, but that cannot be grown or identified through the PCR?
Thank you for your question. Yes, I agree that both situations are possible. It can either be an unidentified pathogen or leftover effect of a pathogen that was there, but I have a theory on culture negative samples. I believe that these are regular pathogens that cause mastitis, and they are not there anymore. As we can see, if you go back to the graph, the most the mean relative abundance was increased for Streptococcaceae, Enterobacteriaceae, Staphylococcaceae, but since we pull all of these mastitis that we cannot identify a pathogen into one, we cannot pick up neither of these bacterial types. And that's my thought on this.
Thank you.
Thank you.
Okay, I'd like to ask you to join me in thanking Dr. Ganda again for her presentation.